certainly have fears that there is a serial killer at loose in Perth.
Sarah Spears, Jane Rimmer, Ciara Glennon.
And every time you saw a young girl walking by, you think, oh God, is she going to be the next victim?
Now, one man stands accused.
If police are right and Edwards is the Claremont serial killer, he's been hiding in plain sight for 20 years.
Today in court, screens erected to block sensitive footage came down and props were brought in to show how Bradley Edwards' DNA was extracted. Natalie Bonjolo and Tim Clark joining you for day 35 of the trial, along with forensic scientist Brendan Chapman, who's going to try and explain and simplify some of today's evidence for us. Because, Tim, what you've been listening to is incredibly important to the case, but it's also incredibly complex.
Yeah, both those things are true, Nat. It was an important day, the first day of the DNA portion of the trial, which we've referenced so many times. And right back at her opening address, Carmel Barbagallo, the chief prosecutor, did warn us all that it was going to be dry, but she had to. This is what she has to do to go through all the continuity of this evidence. And Justice Hall did say to her, well, you've got to do what you've got to do. And that's what she was doing today. And so the first witness was a lady called Anna Marie Ashley, who is now, at the moment at Path West, is a scientist in charge of reporting, which is one of the most senior roles at Path West. But back in the day, back in 95, 96 and 97, she was one of the forensic scientists that was brought in to do some of the testing on some of the exhibits that are so important to the trial. And they started today with the exhibits that were taken from the rape victim at Karrakatta in 1995. And all the evidence that went with that, the physical evidence that was collected, and then the DNA and the scientific processes that she went through to extract the DNA that we now know belongs to Bradley Edwards. And so that's where we started today, and at times it was quite laborious, at all times very complicated, but hopefully Brendan will be able to break it down for us somewhat and explain exactly what she was explaining.
Okay, so perhaps before we run through Ms. Ashley's evidence, Brendan, could you give us a brief overview of DNA extraction from swab to a database?
Yeah, sure. So extraction is a small part of that process. It basically, the very first step that we take is trying to identify the origin of a sample. Now, if we believe it to have come from a body fluid of sorts, we've got a number of chemical tests we can do. So once we establish that, we then go on to extract the DNA. From there, we then move further down, I suppose, the pipeline of a DNA laboratory, and we do what's called a DNA quantitation assay or a quantitation test. Following that, because that informs us to then be able to do our PCR, which is stands for polymerase chain reaction, and I'm going to explain this in much better detail as we move on. But and that's where we copy up DNA in order to generate enough material to give us what we can test to identify a DNA profile. Once we have that DNA profile, that's then pretty simple in terms of it's just a series of numbers that can sit in an Excel sheet, and that can be uploaded to a database and searched against. Okay, so with that in mind, Tim, can you maybe start with the start of the evidence today, the Karrakatta victim, the swab. Where was the swab taken from? Where was that analysed, and how was that done?
Yeah, so as we've explored already during this portion of the trial, the victim at Karrakatta went to get some help, found that help at Hollywood Hospital, just across the road from where she'd been attacked, and was then taken into the custody of police, who did various things. One thing is they took her back to the scene where they discovered her shorts and other items that had left behind. But then she was also taken to the sex assault squad, basically, where she was examined. And during that examination process, some intimate swabs were taken, which were then became part of this suite of physical evidence, which was then taken by the police to the Pathwest lab. There were 23 exhibits in all that were taken to Pathwest, and including in those was a variety of intimate swabs that had been taken from the victim. Some days later, or over the next few days, as Brendan's indicated, some initial testing was done on some of those, and on one particular swab, there was high markers and significant markers for both blood and semen, and so that swab then became one of the main exhibits that they concentrated on at Pathwest, but that didn't happen until some months later, in fact, the following year. So we know the rape was in 95, then in 1996, Miss Ashley was the scientist who was tasked with actually extracting the DNA from this swab, and she described in meticulous detail how that was done, which basically started with the cutting of the swab with a scalpel and then all the scientific processes that followed that.
So, Brendan, if you can just talk us through this part, cutting the swab, how does that happen and where does that go to? Is it put into a test tube or what happens next?
Yeah, so once we have a swab that contains the material on it, that's pretty much our only opportunity to identify what might be on it. And as Tim's alluded to there, they've done testing and identified that there was a result indicating blood and a result indicating semen. Once we've got that result, we then basically will destroy the swab in the next series of processes that we go through. And that's where you've heard the story of chopping up the swab. So if you can imagine, and I think I've explained before, a swab looks like kind of the cotton tips. You need to excise the end of that cotton tip away, and that then goes into a tube that's mixed with some chemicals, basically. What those chemicals do is break open the cells that are contained within that swab. So if you imagine, I suppose, a stylized cell as looking like a fried egg, which is probably the one that most people have seen from high school sort of textbooks, you've got the yolk in the middle. That yolk is where the DNA lives. And it's actually quite robustly packaged up in a cell in order to protect it, which is what we want. We want our DNA protected. So we need to get into it. And so that's where the chemicals come into it. And we use these chemicals and other techniques such as heating to break our way into that egg yolk to release the DNA into the solution. So that's why you're hearing of this swab head cut off, chopped up, put into a tube, then these reagents, these chemical reagents are added that do that.
And Tim, from that point, was there then discussion about, the separation of the cells and that kind of detail?
Yeah, very much so, Nat. So as Brendan's just explained, and Miss Ashley explained in court, at the end of that process, they were left with two physical exhibits, I suppose. One with what she called a sperm fraction, and one which was, I think, Brendan, correct me if I'm wrong, the epithelial fraction, and it was obviously the sperm fraction which was the important one, or the one that was concentrated on later on, because other processes were done in the months that followed to amplify that those fractions, and then do tests on them to basically extract the actual DNA, and so they could get a DNA profile, which was the next portion of what Miss Ashley explained that she had done.
Brendan, do you want to maybe just explain the epithelial cell, what that is, what we're talking about here?
Yeah, so you're correct there, Tim, in the terminology used. When I explain the fried egg look, that is really what most cells in your body look like, and most of the cells that we deal with in forensics are largely epithelial cells. These are the cells that make up your skin, all of those sort of linings, I suppose, throughout your body, and they're that traditional fried egg looking cell. As you know, as most people would know, a sperm cell doesn't look like that. A sperm cell looks, for a better description, like a tadpole, I suppose, and has some unique characteristics about it that are different to the fried egg cell, the epithelial cell. Now, if you remember when I spoke about putting those into that chemical cocktail, we can exploit those characteristics of the different cells in order to pop them open at various different times. So sperm cells are actually much more robust than epithelial cells. So what we can do is we can actually use a lighter or a more gentle, I suppose, chemical, pop open all of the epithelial cells and those only, remove those, and then use a harsher chemical on those sperm cells to pop them open. What that gives us is what you're hearing, which is the sperm cell fraction and the epithelial cell fraction.
Okay, so with the fraction, what happens next, or did they explain what happened in court next, Tim?
Yeah, so after all those samples were extracted and separated, they were all given particular identifying numbers and letters. And the sperm cell fraction is the important one. That was given the identifier of 11J7, which referred to the number of the actual box that it was placed in, which was box 11, and then J7. If you imagine an old-style map grid, it was that's the square, or that's the portion of the box that it was kept in. So this is in 96, and then in 97 there was more testing done, which hopefully Brendan will explain to me, as everyone, along with everyone else, which is called the CTT triplex test, which I think was an amplification model or a more advanced DNA sampling anyway, and that was done in 1997. And from there, that was when the very solid male DNA profile was obtained, along with the sample from the victim, which was obviously different because it was female, not male. And then these were entered into the database. And as we know, the identity of that male DNA was not known for more than 20 years, but it did become very important in the whole macro investigation because it was in that database. And then when other DNA samples, particularly the one from under Ciara Glennon's fingernails, was run many years later, there was a subsequent match.
So, Brendan, can you, as Tim was just explaining, can you talk to us about that process where the further testing was done and the CTT triplex?
So once we've got our DNA extracted out of the cell, that's when we can now manipulate it in order to analyse it. We still, and in all DNA analysis, pretty much across any technique, whether you're studying cancer or undertaking forensic tests, what you need to do is what we call amplify up how much DNA you have, because natively there's just not enough of it. So the best comparison we can use to explain how this works is using a photocopier, where you start with one copy and you put it into a photocopying machine, and imagine you had the photocopying machine set to 2. So you copy off 2 copies, and then you grab those copies and you put those back into the feeder and you copy it again and you end up with 4. And then you take the 4 and you end up with 8, and so on and so forth. So we go through this doubling technique in the laboratory, which is called PCR. And you're going to hear all about PCR throughout the case, and various techniques for PCR, but this is polymerase chain reaction. And this is a well-established technique in molecular biology where we copy up the DNA, and then once we've got our kind of stack of photocopied DNA, we then have enough to analyse it, which in this case, this test is quite an old technique, but you're essentially looking at 3 areas within the DNA to identify individualities between one person and another. To give you some perspective, that was 3 areas that were looked at then. Shortly after that, we started looking at 10 areas of DNA, and now we look at in excess of 20 areas of DNA. So this is kind of where we've come since 96.
And there was a lot of talk, wasn't there, Tim, of genotyping. Is that still a current thing?
Yeah, genotype is just a word used to explain really the result that we're getting. So a genotype is an explanation of what the DNA is. So in the case of this test, it might be 3 pieces of information. In a more modern test, there'd be 20 pieces of information. Those pieces of information give us what's called a genotype. It's like a blueprint, I suppose.
Yeah. Tim, I guess the question people might be asking is, why is it so important that the prosecution outline these processes and procedures at such great detail?
Well, multiple reasons, Nat. I mean, firstly, they've got to prove to the court that the processes they went through were solid enough to get proper results, obviously, because if the results are errant and the DNA doesn't in fact match, then the whole case is at risk. The second is the solidity of these processes is also being challenged from a continuity standpoint. So who touched these samples? How many times were they touched? And most critically, was there any chance that any of these samples, when they were being processed, could have been contaminated either by one of the scientists doing the tests or, more critically, from another sample? Because that's where that's where the defence is going with its line of questioning. And thirdly, it's an interesting history lesson, I think, in how far DNA technology has come in a relatively short time. As Brendan explained, this CT, and was explained in court today, this triplex test only pointed to or looked for a finite number of areas. And the modern test, which I'm sure Brendan will be able to explain, is the PowerPlex 21 test, which is the one done now, looks at a lot more areas. And the very last thing that we heard from in court today, we saw for the first time in public Mr. Edwards' DNA profile flashed up on the screen, and Miss Ashley was asked to point out the areas on this triplex test that matched the modern day PowerPlex 21 test, which was done after Mr. Edwards was arrested, and she pointed across the 2 tests and showed where they matched exactly, and then she was asked to do the same with the DNA profile of the Karrakatta victim as well. So it was a long time getting there, but right at the end of the day, that's where they were leading up to, is that all this DNA testing had extracted this unknown male DNA, and then Miss Ashley was right at the end of the day able to point to Mr. Edwards' DNA and say, yes, that bit matches, and that bit matches.
Was the witness asked specifically about the possibility or any evidence of contamination?
Yes, she was, and that was the denouement of her questioning, as well as the showing of this DNA profile. She was asked during the testing, the original testing, did anything extraordinary happen? And Miss Ashley said no. And then she said, reviewing all those test results from all those years ago, is there any sign of any contamination from another sample or from different samples. And again, she said there was nothing there that indicated there was. That's where we left off today. But Miss Ashley's got a fair way to go in her evidence. She'll be back again tomorrow.
This raises an interesting question from one of our listeners who's got a question for Brendan, which is, in the case that a particular sample is contaminated with DNA other than Bradley Edwards, can it be determined that there was a greater quantity of DNA that belongs to that person as opposed to the other person who's got some DNA there as well?
That's a really good question. Yes is probably the short answer, because when we look at a DNA profile that contains DNA from more than one person, and we can start to work out who has contributed what components of that DNA, we get an indication of how much signal is there, or how high that resolution is, I suppose. And so, for instance, if you have signal from person A that is twice as high as signal from person B, you can infer that there's twice as much DNA from person A there than person B.
Another question is, if one DNA sample contaminates another, what happens to the resultant sample?
Yeah, you would expect to see basically a mixture of both of those individual samples. So if sample A contaminated sample B, whatever is in sample A, whether that's one person, two people, whatever the source is, you would expect to see that in then the subsequent sample B.
That explains it.
Cool.
I mean, it's very complex, and we're not scientists, and it must be very difficult for everyone, including the legal teams who are also not scientists, and you know, I guess people are trying to understand information which takes years and years of university study. How hard would it be for a layman to be understanding this?
I guess that's why we don't have a lay person's jury in a case like this. And it's also why we actually see a bit of a trend for a lot of keen students to undertake both law and science studies, because I think it is a really good double-barrel degree.
Yeah, and I am a layman, Nat, and I can tell you I was struggling today to keep up with some of the explanations. And I've got to say, Miss Ashley was a really expert witness. She was explaining it in such a way that we were, the journalists in court, were able to grasp it just. But it's an interesting topic that Brendan raises there, because DNA is more and more central to more and more cases. The senior counsel who run these type of cases do become quasi-experts in it, and there are, I know of a handful in Perth that really know their stuff on DNA, because they have to, and that goes for defence lawyers as well, that almost specialise in DNA cases, because it is such a boom industry in the legal industry, in legal circles, I suppose. But yeah, it is very complicated, but Miss Ashley was able to give her evidence in such a way today that we grasped it, and certainly Justice Hall was asking all the pertinent questions that you would expect of him, but was also asking some that had occurred to me as well in terms of general process, what titles were being used for certain processes as well, so he could get it all clear in his mind. And maybe come back to me in a few months to see how much more expert I am. But
I'll have an exam for you, Tim.
Oh well, yeah. I think I'd have a good shot at passing it if day one of the DNA evidence is anything to go by.
Do lawyers and the likes come to you for a crash course if they really do feel that they need this additional knowledge?
The DPP has a really close relationship, obviously, with the other government departments, and there's ongoing professional training, I suppose, professional development that they undertake. I'm not sure in the way of defence lawyers. We occasionally get approached by defence lawyers for expertise to assist them in sometimes just clarifying a report. In other cases, they want us to have a much more involved role in I suppose rebutting a prosecution report. But the thing is, at the end of the day, the science is pretty sound. The laboratories are doing things with all due process. If I was to comment on a prosecution report or a report from a government agency, in most cases, I would agree with it.
Yeah. Tim, will Miss Ashley be cross-examined tomorrow?
Yes, eventually. So her first, the next portion of her evidence in chief will be her involvement in the extraction of the DNA from Miss Glennon's fingernails. She was very involved in that, as was Laurie Webb, her colleague, at the time. And so that's where we'll get to first thing tomorrow. She was involved in that process of, I think, swabbing those samples. There was some extraction done at the time as well, and so obviously that's absolutely vital to both sides of the case. So that's where we'll get to first thing in the morning. How long that will take, we're not entirely sure. But yes, eventually, obviously, she will be cross-examined, and that will be very important to both sides too.
Yeah, I mean, this will be critical because this is where the defence will want to show that DNA at some point came into contact or became cross-contaminated with Ciara Glennon's fingernail exhibits.
Yeah, well, they'll try to get there, and they'll try to unpick some of the processes that Ms. Ashley went through today. Because the storage of the exhibits that we've been talking about today is also obviously vital, because that is where Mr. Edwards' DNA was first introduced to Path West, because obviously all the processes that we talked about today happened in 1995, 1996, and some of them in 1997, but the original ones were also obviously before any of the murders had taken place. So yeah, that's where we'll get to, and that's where Mr. Jovich and Genevieve Cleary, his offsider, will no doubt head when they get their chance to cross-examine.
All right, well, before we go, we do have a question for you, Tim, which is back to your area of expertise as opposed to forensic science. Is it permitted for some of those unanswered questions, for example, when Edward's first wife's partner said, "I feared for my life," to be answered, as in, can reporters externally seek out the answers to those lingering lines of questions that aren't solved in court, particularly as there is no jury that could be swayed by reporting?
Yeah, that is a good question, and the answer is yes. Once a witness is allowed to leave court, that is they are dismissed as a witness or excused. That is their involvement in the case done. There can be times when a witness will be excused, but not totally excused, so they can be recalled. But for instance, in the example the questioner uses for Mr. Edwards' wives, for instance, they would be now free to talk to us if they wished to. Now, whether we would publish that material while the trial is ongoing, that's another question, and that would be an editorial question that we'd have to weigh up. But as the questioner also points out, there is no jury in this case, and so it's basically legally accepted that a judge cannot be swayed by media reporting one way or the other in terms of a verdict, whereas a jury obviously can be. So yes, we would be at liberty to go and search out Mr. Edwards' wives and any other witness that had finished from now. But to be honest, we're so busy reporting on the day-to-day that I'm not sure that has actually taken place. But once the trial winds down, I can assure you that myself and many other of my media colleagues will be searching out certain witnesses to see if they want to give us some more information.
Yes. Thank you both for today, and thank you for joining us for day 35 of Claremont in Conversation. I'll be back tomorrow with Tim and Alison Fan to wrap up week 8. Chat to you then.
This podcast was produced by Kate Ryan and Alicia Preedy and recorded in the studios of 7 West Media. Audio files were provided from the archives of the 7 Network and the West Australian. Sign up for daily emails and all the latest on the Claremont trial at thewest.com.au.
Enjoying this podcast? If the story behind the headline matters to you, then you can count on thewest.com.au to deliver. For more on Claremont the trial, follow the live blog, watch the nightly news updates, and sign up for daily email updates at thewest.com.au. Subscribe now for just $1 a day at thewest.com.au.